This video will cover the anatomy and function of the eye, a crucial sensory organ. We will begin by looking at the external appearance of the eye. Key eye functions include transmitting and refracting light, converting light energy into action potentials, and sending these signals to the brain. The eye utilizes a transparent light path and structures that bend light, known as refraction. The eye is composed of several layers and chambers. The layers are the fibrous, vascular, and nervous tunics. The chambers include the anterior, posterior, and vitreous chambers. The ocular segments are defined as the posterior segment, which includes the vitreous, retina, choroid, and optic nerve, and the anterior segment, which comprises the structures in front of the vitreous, the cornea, iris, ciliary body, and lens. The tear film has 3 layers. The oil layer, from the meibomian glands, prevents evaporation. The water layer, from the lacrimal glands, lubricates and washes away debris. The mucin layer attaches the tear film to the eye and spreads water evenly. The cornea is transparent, transmitting and refracting light with a power of +4O to +44 diopters. It is avascular, and its relative dehydration is maintained by endothelial cells. The cornea's anatomy includes the epithelium, which serves as a barrier, the stroma, composed of collagen and keratocytes, and the endothelium, which maintains dehydration. It is densely innervated, making it the most sensitive organ, and protects the ocular interior. This slide illustrates the structure of the crystalline lens. The crystalline lens is made of water-soluble proteins called crystallins. It transmits and refracts light, contributing +17 diopters of refractive power. It also enables variable light refraction through accommodation. Accommodation allows for clear vision at varying distances. For far objects, the ciliary muscle relaxes, the zonules tighten, and the lens flattens. For near objects, the muscle contracts, the zonules relax, and the lens increases in convexity. For a visual explanation of accommodation, please search for accommodation reflex on YouTube. Emmetropia is the state of normal cornea, lens, and eye length. Myopia occurs when the eye is long, and hypermetropia occurs when the eye is short. Retinal function involves photoreceptors converting light into action potentials. Cones provide high-acuity color vision in light-adapted conditions. Rods provide sensitive, low-resolution vision in dark-adapted conditions. Signals transmit through bipolar and retinal ganglion cells to form the optic nerve. The spectral sensitivity ranges for cones include S, or blue cones, M, or green cones, and L, or red cones. This image shows the retinal microstructure. Here is another view of the retinal microstructure. The fovea, which is central to the macula, is highlighted in this retinal microstructure image. This image shows a cross-section of the retina. The image is labeled with temporal and nasal regions. This slide illustrates a superior retinal detachment. This is a microscopic view of the optic nerve head and adjacent retina, stained with Masson's trichrome. The ciliary body facilitates accommodation via smooth muscle and is the attachment point for zonules. It secretes aqueous humor, which nourishes the cornea and lens and maintains intraocular pressure. This is an image showing ocular structures. Here are some of the ocular structures. The uveal tract is the eye's vascular and immunological pool, consisting of the iris, ciliary body, and choroid. The iris regulates the pupil size, the ciliary body manages aqueous humor and accommodation, and the choroid, the most vascular tissue, nourishes the retina and sclera. Eyelids and conjunctiva protect the eye. Eyelids distribute tears, cover the eyes during sleep, and house the meibomian glands for the oil layer. The conjunctiva attaches the eyeball and permits rotation, clearing debris. This image provides another view of the eyelids and conjunctiva. The orbicularis oculi muscle is critical for eyelid function, with 50 fibers in the upper and 25 in the lower eyelid. Eye movements include adduction, abduction, depression, elevation, intorsion, and extorsion. Key structures related to extraocular muscles include the annulus of Zinn and the trochlea. The 6 extraocular muscles are the medial rectus, lateral rectus, superior rectus, inferior rectus, superior oblique, and inferior oblique, each responsible for specific eye movements. Extraocular muscles are innervated by 3 cranial nerves: the abducens nerve for the lateral rectus, the trochlear nerve for the superior oblique, and the oculomotor nerve for the other 4 muscles. For a detailed video on extraocular muscles, search for Extraocular Muscles Eye Anatomy on YouTube. Tear production starts at the lacrimal gland, and drainage occurs through the canaliculi, leading to the lacrimal sac and duct. This is another image of the eye and its associated structures. The visual pathway starts at the optic nerve, moves to the optic chiasm, then the optic tract, the lateral geniculate nucleus, the optic radiation, and finally the primary visual cortex. This diagram illustrates the effect of injuries at different points along the visual pathway, from the optic nerve to the primary visual cortex. This is another view of the visual pathway. Cataracts of the crystalline lens are classified by age of onset, location, such as nuclear sclerotic or cortical, and cause, including age-related, traumatic, or diabetic. Phacoemulsification is a surgical technique for cataract removal. It involves capsulorhexis, phacoemulsification of the lens, and then the injection and placement of a foldable intraocular lens in the capsular bag. Intraocular lenses can be multifocal to correct vision at various distances, or toric to correct for astigmatism. This scene is blank and contains no content. In 1948, Dr. Jeffrey Orbell, an ophthalmologist and ENT physician, led an expedition near Lake Teanau that rediscovered the living takahe, long thought to be extinct. This concludes the video. Material contained in this video is copyright of the Department of Ophthalmology, New Zealand National Eye Centre, University of Auckland.